This is Chapter 12's lecture. It's primarily going to be over skeletal muscle, so it should correspond with Chapter 12, sections 12.1 and 12.2. Skeletal muscle are the muscles that are utilized by the body for movement. They're usually attached to the bones by tendons, and each muscle will have an origin and an insertion. The origin is the closest part of that muscle to the trunk or to the more stationary bone, and the insertion part is going to be the more distal or the more mobile attachment. Furthermore, muscles can be divided into two categories, flexors or extensors. So flexors, they bring bones closer together. An example would be the biceps that brings the upper arm and the lower arm together when it contracts. An extensor is going to move two bones away from each other. So again, the upper arm, lower arm, the humerus, and versus the ulna and the radius are going to be moved away from each other by our tricep. In this way, muscle groups can be classified into what we call antagonistic muscle groups, where we put a flexor and extensor pair together, so the bicep and the tricep. All skeletal muscle is comprised of connective tissue that holds the muscles together into the fascicles, as well as holds it onto the bone or around the bone in its various spots in the body. We have blood vessels and nerves that are going to run through that skeletal muscle, and then we have the actual skeletal muscle itself, what we call the muscle fascicle. So we have lots of muscle fascicles that are grouped together into a single motor unit. If we break it down to the smallest functional unit of the muscle fascicles, we would get a muscle fiber. That's a single cell. Each muscle fiber then will contain sarcoplasm, which is what we call the cytoplasm inside. It'll have multiple nuclei. It'll have what we call the sarcolemma, which are these T-tubules that make up the outer membrane of the muscle fiber and are linked functionally to our sarcoplasmic reticulum. And the sarcoplasmic reticulum would be analogous to endoplasmic reticulum in a normal cell. We then have mitochondria. We have glycogen granules that hold the glycogen that we'll utilize for energy. And then we have what we call the contractile elements. These are the myofibrils. And our myofibrils kind of get broken into three groups, right? We have those that are associated with what we call the thin filaments, and it's the thin filament because when we look at it under a microscope it looks thin. And then we have those that are associated with the thick filament, and we have some accessory proteins. So we'll talk about each of these, and you're responsible for knowing the function of each of these fibers. So the thin filament is primarily composed of the protein actin. Associated with actin are two additional accessory proteins called troponin and tropomyosin, and we'll talk about what they do when we get to muscle contraction. The thick filament is comprised of what we call myosin, the protein myosin, and there's two accessory proteins associated with both the thin and thick filaments. Those are titin and nebulin. So these make up all of the myofibrils, and the arrangement of these myofibrils is what we call it being organized into the sarcomere. So let's look at sarcomere organization really quick. So over here you see this is what a muscle fiber would look like under a microscope. So we're looking at these muscle fibers, and you notice there's these different regions associated with density of the proteins found there. So here's a relaxed muscle, here's a contracted muscle. And so we have the organization of the sarcomere has been identified based off of the microscopic images, right? So what we see first off is that we have these regions that seem to anchor all of our myofibrils. We call those the Z discs. So on either side we have the Z disc, and then we see this region in which it's lighter. Okay, this is what we refer to as the I band, and in this region the only proteins that we're seeing is primarily actin, right? So we see this actin right here. And of course, with actin, there's going to be the associated troponin and tropomyosin. Then this next region that we see is this darker region. Okay, this going from this region, the darker region, all the way over to here, we refer to as the A band. Okay, it's dark because we have both overlap of our thin filaments and our thick filaments. And you'll notice right here in the middle, we have the middle line, or the M line, that everything is anchored to. And then we have this region in which we get a little bit lighter. This is where we would only have myosin. So here we have myosin and actin overlap. Here, in a relaxed state, we can see there's a stretch in which we have just the thick filaments. This is referred to as the H zone. Okay, so H zone, A band, I band, Z discs, and the M line. What was also noticed is that during contraction, the H zone disappears, right? And what that tells us is that we now have our actin and myosin. The actin is sliding across or over the myosin, so we have overlap. And you'll notice the Z discs, they come in, they're a little bit closer. So we're contracting this muscle, and really it's this overlap between myosin and actin that gets filled in, right? So our I band isn't quite as big as it was here because we have more overlap between our actin and our myosin. So based off of these observations, we started to develop something called sliding filament theory. Now, I told you I would talk a little bit about some of our accessory proteins. We'll get to troponin and tropomyosin in a minute, but let's talk about titin and nebulin. Okay, so titin gets associated with our myosin, our thick filament, and what it does is it helps anchor. So myosin is anchored onto the M line, and then titin anchors it to the Z disc. So it provides elasticity and also stabilizes this molecule so that when the thin and thick filaments are sliding past each other, they don't move out of alignment. Nebulin is associated then with our actin, and it's going to help, again, align the actin and keep it in the correct position with our myosin molecule. When we contract our muscles, we are generating a muscle tension. As those filaments slide past each other, the force that's created by that muscle we call the muscle tension, and with that force, we then can provide energy to lift a load. So a load would be the weight or force that's opposing that contraction. So every reaction has an equal and opposite reaction. If we create a tension, then there must also be a load that forces back. So if I contract my bicep when I'm doing a arm curl, right, the amount of weight that I am able to lift, that is the load that is counteracting the tension I am generating by contracting my bicep. So when we contract the muscle, that's what causes that tension in the muscle, that sliding filament theory, in which we have the actin and the myosin sliding past each other, and we're shortening the muscle fiber. That creates the contraction. Relaxation would then be the release of that tension when those filaments slide back and forth. So if we break down what muscle contraction, the different points of muscle contraction would lead to muscle contraction. There's three things. One is there's something that's occurring at the neuromuscular junction to initiate. So that would be the initiation of it. Then we have what we call excitation and contraction coupling. So how that neuromuscular junction spark then leads to the muscle starting its contraction. And finally, we have that sliding filament theory of how the fibers themselves move to generate that contraction. So let's look at all this. I explained some of this when we were looking at the actual microscope slide of the muscle themselves, but again, the support for sliding filament theory is seen when we looked at a relaxed muscle versus a contracted, and how that H zone is reduced and the I band is reduced as well on both sides. And so that tells us that when we're contracting, we must have these two filaments, the thick filament and the thick filament, sliding past each other. So how does that occur? Well, if we look at the fibers themselves, what we see here, this is our actin and here's our myosin. So the myosin, all along the myosin protein, there are these active sites that we call myosin heads. So this is part of the protein, and we have these heads that have a binding site that can bind to actin. On actin, we have those two additional accessory proteins, troponin and tropomyosin. Okay, these are going to be important in the relaxed state because tropomyosin, it's going to block the active site of myosin. So I like to give an example when I talk about this sliding filament theory. Let's say you and your friend have gone out to a club to spend a night out. Okay, and you are actin, and your two friends, one is troponin and one is tropomyosin. And one of your friends is there to make sure that no boy or girl talks to you, right? They're keeping everybody else away because they're like, no, this is a girl's night or this is a guy's night. Pick the side that you're on, right? And we don't want anybody else interfering. Okay, so they're blocking, in this case, potential mates, potential guys or girls, which would be the myosin head, from interacting with you. Now that's a weak interaction. Now what happens when we talk about muscle contraction is one of the things that changes inside the cell is the amount of calcium. So troponin right here has a binding site for calcium. So when calcium levels increase inside the cell, it's going to bind to that troponin. So again, using my club, let's say that calcium is the music and one of your friends loves music. When the music starts getting loud and starts to play, they go, I love this song, I'm going to start dancing, right? And they pull their other friend, tropomyosin, to come dance with them. You got to come dance with me. Now that leaves you unguarded, right? And the potential mates, whether you're a girl, the guys, or if you're a guy, the girls, can now come and interact with you. And what happens in this case is the myosin head, it has ATP bound to it. So if we come back here, you see when we take ADP and we cleave off one of the phosphates, right? We make it AD. This is diphosphate instead of triphosphate. Adenosine diphosphate. When that phosphate gets cleaved and it's leaked down, we have this weak attraction. So basically, it's like the guy or the girl is checking you out, right? He then leaves his group of friends. He jettisons out that phosphate and says, I'm going to interact. Now we have what's called the power stroke, where we have this tight bond. They come up to you and they're engaging you in conversation. They're holding your hand and saying, hey, let's dance together, right? That's this tight attraction, okay? And at that point, we're in what we call the rigor state, right? We're tightly bound to the actin molecule. Then someone comes up and puts some energy back onto the myosin friend. So say one of his friends, if we're talking about, let's say actin is a girl, myosin is a guy, one of his friends comes up and punches him in the arm and says, hey man, what are you doing? Right, that's putting energy. That causes a release. Okay, so we release as long as that punch is there, he's not going to be activated. But as the punch starts to dissipate, right, we cleave off the phosphate and we get that next hydrolysis and weak contraction. It comes back in and we're moving. So we then get what we call this power stroke, in which we are continually grabbing the actin molecule and pulling it out. Now, this is where I have to vacate my analogy of a club and the two friends, troponin and tropomyosin, blocking guys from meeting a girl. And now I want you to envision actin is like a rope, okay? And you have a rope, you're trying to pull that rope. If you're trying to do it by yourself, and let's say you only have one hand, right? Every time you grab that rope and you pull it towards yourself, you're creating some tension. But as soon as you let go with that one hand, what's going to happen? The rope is going to slip away. But if you have two hands, you can do this hand over hand action. You grab with one hand, right? That's when we grip it hard, that's when we have just ADP. Okay, that causes a conformational change in this head where we cock it, right? So we kind of pull it closer to us. ATP then gets put into it, that would be the release. But because we have multiple hands, this actin molecule has lots of heads. We're just seeing a single head here, but there's actually lots of heads at play. And so it's hand over hand that we're pulling this in. So you grip, pull, release, and then another hand is able to grip, pull, release, and it kind of, you move that way. And that's how we slide the filaments across each other. We call that the power stroke. It's contraction and relaxation of those myosin heads to then create that tension. Okay, these figures are really great in your book that explain the power stroke and the relationship of ADP, ATP, and what it does. Okay, it's also a great explanation of why we have, say, our rigor state. So if you remember, when we're bound, that's the rigor state, and we're bound when we have no ATP, right? ADP. So when someone dies, at first their muscles are probably going to be relaxed because they haven't initiated a power stroke. But over time, what happens to the energy stores in our cells? Okay, if you die, your cells function for a certain amount of time, but at some point they're going to use up all the ATP, and then they're going to die as well. When that ATP in your skeletal muscles is all lost, all we're left with is ADP. As the cells also start to deteriorate, some of that calcium that's being stored is going to spill out into the cell, and that's going to move the troponin. It's going to bind to troponin, move the tropomyosin out of the way, and all the myosin heads are going to lock on. They'll not release because the only way they can release is if we have active ATP. The body's dead. It can't generate any more ATP. Hence, we have rigor mortis, right? Where a couple hours after a body dies, it becomes stiff. The muscles lock up because they all contract and it's hard to move. What triggers this? Okay, we talked about excitation and the power stroke, but let's talk about initiation. So we have our somatic motor neuron. It's depolarized. It has an action potential. When that action potential meets the axon terminal, it's going to depolarize it, triggering exocytosis of acetylcholine. Okay, so that acetylcholine gets released into the motor end plate. And in our motor end plate, we have a bunch of sodium channels. Okay, those binding of acetylcholine to those nicotinic receptors are going to open up sodium channels, and sodium is going to enter into the cell, which depolarizes the membrane of our muscle fiber. That causes an action potential that moves down the membrane into what are called these T-tubules, where they kind of innervate deep into the tissue. And at that T-tubule, we have these couple of gates, right? So we have our dihydropyridine calcium channels, and inside the cell, so right here is our sarcoplasmic reticulum. We store calcium in the sarcoplasmic reticulum. And you'll see how our DHP gates and our ryanodine receptor channels in the sarcoplasmic reticulum are connected. So this depolarization triggers the opening of our RYR channels, and we release that calcium into the cell. Okay. Now, as the calcium gets released, what does that do? Again, going back to my analogy, that's like turning on the music. Troponin, I love that song, says to tropomyosin, because those two are attached at the hip. They never leave each other's sides. Come dance with me. Now actin is accessible and open for the myosin heads to hit on them, which causes our power stroke. So that's the excitation contraction coupling. We had initiation, excitation of the calcium, and then it gets coupled with our power stroke. What do we do at the end? How do we relax the muscle? Well, relaxation, it occurs by pumping calcium back into the sarcoplasmic reticulum. So you're turning the music off. We use a calcium ATPase pump. Okay, so we have to use energy. We push calcium against its concentration gradient, in some cases back into the sarcoplasmic reticulum. As calcium concentration levels decrease, it disassociates with troponin, which moves tropomyosin back. It blocks the myosin heads to where we start to lose the number of hands on our rope, and it starts to slip back into its relaxed state. The excitation coupling of when we have the muscle fiber being innervated and then we generate that contraction. It starts at the end plate, in the motor end plate, and we generate what's called a end plate potential, or an EPP. And if we look at the amount of time that occurs between the somatic motor neuron having its action potential and when we actually get the contraction, we see that there's a little bit of delay, right? So here's the action potential. Here is the muscle fiber action potential that occurs. We're traveling down the T-tubule, so there's a little bit of time there. And then we actually have the contraction and relaxation. So that period of time between when the somatic motor neuron releases its neurotransmitter or gets triggered to when we actually start contraction, we call that the latent period. So there's a latent period. The time between when we contract and relax, this is called a muscle twitch. Okay, so we have this twitch. Our latent periods, it is a little bit of a delay, and the reason for that delay is basically the amount of time it takes to transmit the depolarizations across the membranes and the amount of time it takes for calcium to be released into the sarcoplasm. In general, each one of your muscle fibers has enough ATP for exactly 8 twitches, which you may say, that's not a lot of energy, Dr. Smith. Like, I know I can do way more than 8 reps when I'm lifting weight. And that's true. And the reason for that is this is how much ATP is already generated. But the body can make more, and we have a backup energy source. Really, what we need to do is we take those ATPs and we keep rephosphating them. So that backup energy source is phosphocreatine. Phosphocreatine is this molecule that gets converted. Basically, we have a creatine model right here, and we can stick a phosphate onto it utilizing creatine kinase, this enzyme, and now we have an extra phosphate stuck onto it. So we take creatine, we make phosphocreatine, and then when we need to re-energize our EADP, we can take that phosphate back off and stick it onto the adenosine diphosphate to make ATP. So we're constantly storing phosphates on our creatine. So when we're at rest, we take all that ATP from metabolism that we're generating from glycolysis, from the Krebs cycle in our mitochondria, and we're sticking them into creatines, and we're creating lots of ADP and phosphocreatine. Then when we start working, we use creatine kinase in reverse, take that phosphocreatine, and then take the ADP that's now being utilized every time we We cleave off that phosphate in the power stroke so that we can attach. We stick another phosphate back on from our phosphocreatine. Okay, so and that's the process that we kind of go through over and over again. Can we ultimately run out of ATP? Sure. If you no longer have glycogen that we're utilizing in our glycolysis, in our Krebs cycle, at some point the cell would not be able to create any more ATP, and we wouldn't be able then to have a relaxed muscle. Again, when we die, that would be rigor mortis. But the point I also want to point out is we generally don't store a ton of ATP. We don't. It's a high energy molecule. We only make it and utilize it when we want to use it, almost immediately. So this is how we can have a lot more twitches, right, than just 8. That brings us to this concept of muscle fatigue, right? You can't go forever. At some point, we are going to have fatigue in a muscle. Fatigue is a reversible condition in which our muscles aren't able to generate and sustain the expected power output that the load is placing on it. And it's really variable as to why we experience fatigue. It can occur because of the intensity of the activity. If we're really doing something that puts a lot of strain, it might occur a lot faster. If we do it for a long time, it doesn't have to necessarily be an intense activity, but if you run a marathon, even though we're not sprinting, you're going to get muscle fatigue. How much oxygen is available in the working conditions, whether we're using aerobic or anaerobic conditions to generate our ATP, is going to influence how quickly we have fatigue. The muscle composition of your skeletal muscles plays a huge role in whether we have muscle fatigue as well. We'll talk about that in a minute. And then your overall fitness. So we can take fatigue and the mechanisms that generate fatigue, and we can classify them into two kind of categories of fatigue, what we call central fatigue and peripheral fatigue. Central fatigue is psychological. These are things that occur in the central nervous system in our brain that tell us we need to stop. It's when you start sprinting or let's take a different example. Let's say it's when you're jogging for a long time, and at some point, it's not the muscle ache, but it's that thought in your head of I can't go on. I really need to stop. It's like that runner's wall that you hit. That's central fatigue. And that's basically the brain saying, stop firing these motor neurons. Let's just stop. Peripheral fatigue, this is all occurring in the muscle itself. So either at the neuromuscular junction, that excitation contraction coupling, or the calcium signaling inside of the cell. And there's a lot of things that can cause this. One, we could be lowering the amount of neurotransmitters that are getting released from the neuron. The receptors could be getting deactivated. That could cause, this peripheral fatigue. The muscles themselves, the membrane of the muscles could become hyperpolarized, and that then makes it harder for us to have that excitation contraction. Then things that actually affect, let's say all of this is working, we could still have issues with our calcium release. So calcium could be leaking out of the cell, which is going to decrease the amount of calcium that comes out of the sarcoplasmic reticulum, and it's going to decrease that interaction that it has with troponin. And then we have a couple other things that come into play that may influence fatigue, and these can be classified into two kind of categories of what we call depletion theories or accumulation theories. So depletion means we're starting to run out of stuff. So we might be running out of phosphocreatine. We might be running out of ATP. We might be running out of glycogen, which generates our ATP, right? Those things would lead to depletion. Accumulation theories now say, hey, the cell starts accumulating things that change how it functions. So one of those, probably the most common, is hydrogen. As we are doing work, hydrogen starts being increased. That changes the pH and the cell functionings of those muscle cells. Phosphate can do that as well. And then the last one is lactate. We used to say, when you felt that muscle burn, that muscle burn is from the lactic acid, which can change pH a little bit. That's true. However, we now know hydrogen plays a much larger role in that burning sensation than lactate. So those are our fatigues. What can kind of cause some of this type of fatigue, particularly whether it's central or peripheral? Well, let's say we are doing an extended, what we call sub-maximal exercise. So I tell you you're in the gym, and I say, let's do a workout in which we're doing low weight, but lots of reps. So we're going to do bench press, but instead of benching 100 and some odd pounds, I say, let's just bench 80 pounds and do as many reps as possible. What's probably going to lead to fatigue in that case, when we're doing sub-maximal exercise, but extended duration, is depletion. It's going to be depletion theory. Generally, we're going to start running out of glycogen in those muscles to allow it to work. Now, if we do short duration, maximal exercise, so now I say, let's put as much weight as possible and see how much you can lift. That is going to usually, the fatigue is going to be caused by those increased levels of phosphate as we burn through to try to get the amount of twitches necessary to lift that load. So we don't release as much phosphate from our myosin as we start to get these increased intracellular levels, and that decreases our calcium release, and we're just not able to do another twitch. Finally, let's look at maximal exercise. So this would be kind of like running a marathon. When we're doing maximal exercise, and I shouldn't just say running an marathon. It's also, you could be sprinting. It's just when it's not as much on what type of load you're doing, but more on a constant state. This tends to be by accumulation of, we start accumulating calcium in the extracellular ECF. That changes membrane potential as we start losing calcium because we're constantly pumping sodium into the cell, calcium out of the cell. We have leaking calcium, or not, sorry, calcium, I meant to say potassium. We're leaking potassium out of the cell, pumping it, trying to pump it back in. That changes the membrane potential. And as that membrane potential changes, our sodium-potassium pump changes. And that makes it harder to continually trigger those excitation coupling reactions. I said another thing that can influence the fatigue that we have is the composition of your muscle fibers. So not all muscle fibers are the same. There's 3 types of muscle fibers that we have. We have what we call slow twitch fibers. They're called slow twitch because it takes a little bit longer for them to generate their tension, but they rely on oxidative phosphorylation. So we're using oxygen. Our slow twitch muscle fibers, because they utilize a lot of oxygen, they're really dark red. And then we also have 2 types of what we call fast twitch fibers. So they develop that tension a little bit faster. They're able to generate ATP quicker and split that phosphate off. They pump calcium faster from the sarcoplasmic reticulum. And the 2 types that we have are what we call our glycolytic fibers, in which we're using anaerobic glycolysis, and then the oxidative glycolytic, in which we're using both an oxidative phosphorylation and a glycolytic, so both anaerobic and aerobic metabolism. And so this table from your book, 12.2, really, really helpful because it explains the differences between these. So in this, for instance, slow twitch, it's the slowest, right? It is the smallest diameter, has but it can contract for the longest amount of time. So you can keep that twitch, that tension for the longest amount of time. The reason why is because it uses that oxidative, that aerobic energy source to generate the ATP. Lots of blood goes into there. It's really dark red because it has myoglobin, lots of mitochondria. So now the next fastest in speed is going to be that fast twitch oxidative glycolytic. So it's much faster, a little bit larger diameter. The duration is short, but it's not as short as, say, our white muscle. This still is going to look red. We mostly use these. So slow twitch is mostly used for posture. These ones are for things like standing and walking. And they're going to, it's kind of like the middle of the road, right? And then the last ones is going to be our fast twitch glycolytic white muscle, okay, in which the color of it is really pale. There's not a lot of mitochondria in there. There's not a lot of blood flow because it's not relying on oxygen. It's entirely glycolytic. So our fast twitch oxidative glycolytic, it's glycolytic too, but when we do endurance training, it'll switch over to oxidative. And we use these things for things like really quick movements, jumping, sprinting, those types of things. The fast movements is what we do. And the fatigue is you can see, this is the most resistant to fatigue, and these are the easiest to fatigue. And so each of us has, based off of genetics, we have different compositions of these fibers built into our muscle fibers. Each contraction, relaxation is a single twitch. But we can have summation, just like we do with graded potentials. There can be summation in muscle twitches too. So if we have a twitch before we fully relax, we're going to generate more tension and more force, which leads to what we call tetanus, right? Single twitch, maximal tension that a muscle can generate is what we would call tetanus. And at some point, fatigue is going to cause that muscle to lose its tension, despite the fact that we still have the neuron saying, hey, stimulus needs to happen, and it's going to phase off. If you don't believe that, try holding your arm out to the side with a 5-pound weight and put your arm directly to the side. At some point in time, that arm is going to start to hurt and be heavy, and you're not going to be able to keep it, and it's going to fall down by your side. Last point we need to kind of talk about is how are our muscles compromised. So I mentioned we have, or comprised. We have 3 types of muscle fibers. Each fiber is composed of lots of different fascicles. And we can have these fibers then get grouped into what we call a motor unit. So each motor unit has the exact same motor neuron. So here in this picture from your book, we have 3 motor units. They color-coded this: red, blue, and purple. We could also think about this as these could be different muscle types, fiber types. So this could be our slow twitch, fast twitch, glycolytic oxidative, and then fast twitch glycolytic. And the reason why I'm saying we could also put these colors with those different types is during embryonic development, what dictates what type of muscle fiber you create is the actual neuron that grows and attaches to them. So that neuron during development is going to start secreting growth factors that trigger the development of the muscle fiber into the type of muscle fiber that it ultimately is going to be. And then it doesn't change. We don't replace these muscle fibers. So it's not like you can generate more slow twitch muscle fiber. You're born with what you have. Now you can train and you can strengthen some of these. You can take a fast twitch oxidative glycolytic, and through endurance training, you can build up its glycogen stores. You can build up more mitochondria in it, and it may function a little bit better like a slow twitch, to where it has longer duration. It still generates contraction fast, but it can last longer. So you can make them more fatigue resistant, but you can't ultimately change the properties of them that much. And so as these motor neurons, develop, all of the axon terminals then create the same type of muscle fiber and they act as a unit. So if this neuron fires, all of the purple are going to twitch. That's why we call it a motor unit. Okay, so the motor unit twitches together, and we use these motor units together to increase load. Okay, I already answered these questions of inheritance and plasticity. The plasticity of our muscles, yes, you can change a few things about the fatigue resistance, but ultimately you can't make a fast twitch, glycolytic, oxidative muscle into a slow twitch. There's not that much plasticity. So how do we utilize motor units? Well, we have what we call recruitment. So if I'm lifting, let's say I'm doing an arm curl and I'm lifting a 20-pound weight, okay? When I do that 20-pound weight, I have to generate enough tension to lift that up. So I'm going to have to probably utilize a couple of different motor units to lift that. If I do 5 pounds, I need less motor units than if I did 20 pounds. We call the addition, that recruitment, or we call the addition of motor units recruitment. So if I start to lift the weight, let's say I was doing 5 pounds, and I'm utilizing, 1 or 2 motor units. And then I switch to the 20-pound, and I start to lift it, and I think it's going to be like the 5-pound, and I realize, oh, this is heavier. So I have to try a little bit harder. That's the recruitment of the brain saying, we need more motor units. Let's put more into that. And that increases our contraction force. Now, if I hold a weight, that same 5-pound weight, out to the side of my body, and I hold my arm straight, and I'm holding it up in the air, how do I maintain that for a certain given time? Well, we start using what we call asynchronous recruitment. So I activate a couple of motor units to hold that 5-pound weight. I don't need to use all my motor units, right? So I activate some, I hold it out, and as they start to fatigue, the body recognizes, hey, this is fatiguing. It innervates another, or it recruits another motor unit and turns the original one off. And so by switching up our motor units, we give them more time to rest and then be recruited again to reduce that fatigue. And so asynchronous recruitment allows us to help avoid fatigue, and we turn off different motor units left and right. Okay, that pretty much covers all the material for skeletal muscles. It's a lot of material. Make sure that you go over the study guide and look at the learning objectives. It outlines pretty well what I want you to know on skeletal muscle. The next part that we'll talk about in part 2 is going to be smooth muscle. Not as much material. It's mostly just comparing smooth muscle to skeletal. The bulk of this chapter is the skeletal muscle.